Hello Chem 213 students, and welcome back for our second video about amino acids, peptides, and proteins. So in the last lecture we reviewed the structures of amino acids and some of their properties, and now we're going to talk about the synthesis of amino acids and the ways in which those amino acids will chain together to form polypeptides. So we're going to go through several reactions that we've learned previously in previous chapters on molecule and saw their effects on molecules that were not necessarily biological molecules. We'll see that these same reactions can be used to produce amino acids. For example, the Hell-Volhard-Zelinsky reaction, which was used to functionalize the alpha carbon on carboxylic acids from chapter 21, can be used to create an amino acid. So we start by using the Hell-Volhard-Zelinsky reaction. We react with a carboxylic acid here with bromine and PBr3 to install the bromine at the alpha position, followed by a workup with water. Finally, then we react with ammonia behaving as a nucleophile, which then replaces the bromine atom in the alpha carbon position, yielding for us an amino acid. So that's one way to synthesize an amino acid from carboxylic acid. We can also synthesize an amino acid from diethyl acetomalonate, and so this is going to be similar to the malonic ester synthesis from chapter 21 as well. So in this structure here, this carbon here does have a hydrogen, and that hydrogen has a particularly high or particularly low pKa, a particularly high acidity, because of the fact that it's alpha to 2 carbonyl groups here. And so if we have a strong base like sodium ethoxide, this is going to then be able to remove this proton, and that's going to leave these electrons right here as a lone pair. And so this makes a carbanion here, and that carbanion can then go and do a nucleophilic attack on an alkyl halide, and the halide leaves as a leaving group, and then we have that alkyl group attached to this diethyl acetomalonate, acetomidomalonate. Sorry, that's a mouthful. And then after that, we can hydrolyze the amide here, and then ultimately get for us the amino acid again. And this would be a racemic mixture of two isomers here, this being the chiral carbon right here. So for example, in this series of reactions here, we'll show how phenylalanine could be made from this acetomalonate, amidomalonate. And again, so we use a base to deprotonate this, making it a carbanion, which then that carbanion, so once we've deprotonated it, we have this carbanion. This does a nucleophilic attack on the benzyl position here, and then we have a benzyl group attached. Finally, we do a hydrolysis, and we remove the ester, and then ultimately produce for us the carboxylic acid. So we hydrolyze the esters and ultimately get our slideshow. Okay, and ultimately get our phenylalanine. So the R side group here being this benzyl group, and then we have the amino group at the alpha carbon to the carboxylic acid. Another way to synthesize a amino acid is it can be synthesized from an aldehyde using what is known as the Strecker synthesis. So for the Strecker synthesis, we react a aldehyde here with ammonium chloride followed by sodium cyanide, which then produces for us an alpha amino nitrile. So alpha amino meaning the alpha carbon of the amine, the one that it's attached to, here contains a nitrile, the C triple bond N. And then finally we can hydrolyze this nitrile here, which gives for us this carboxylic acid. And we still have the amino group, which is now protonated under acidic conditions. And again, that amino group is at the alpha, attached to the alpha carbon on the carboxylic acid. So the side chain of the aldehyde will become the side chain of the amino acid. So for example, if we take ethanol, the side chain here of the methyl group, the side chain is a methyl group, and so when we then introduce the amino group and the nitrile group and then hydrolyze and we get the carboxylic acid, this methyl group that was there is still going to be there. And so this is going to be the amino acid. In this case, from ethanol, we would make alanine, and the side chain would be the methyl group. Here again, we have the amino group on the alpha carbon next to the carboxylic acid. In terms of the mechanism for the aminonitrile formation, so this starts with a nucleophilic attack by ammonia here, and then we have this resonance arrow putting the electrons on the oxygen. So at this point, this nitrogen has 3 bonds to hydrogen, and it has another one to carbon, giving it 4 bonds and no lone pairs for a positive formal charge. The oxygen, instead of having 2 bonds and 2 lone pairs, now has 1 bond and 3 lone pairs, and so it now has a negative formal charge. And so this alkoxide can now be protonated, especially if we react with strong acid like HCl, for example. And so now we've protonated the alkoxy group, and this oxygen is now making 2 bonds with 2 lone pairs, so it has a 0 formal charge. And if we react here with another equivalent of ammonia, this will deprotonate the amine here, and at that point we have the amine and a hydroxyl group. That hydroxyl group can then be protonated. It will be equilibrium, so you see the equilibrium here along all of these arrows. They're all equilibrium arrows. These reactions go, only partial of the way. We react with HCl here, and now we get a protonated alcohol group, and that can now be a good leaving group. It will leave as water. This oxygen has 2 bonds to hydrogen and 1 bond to carbon, giving it 3 bonds and 1 lone pair for a positive 1 formal charge. At that point, we have this delocalized lone pair here on the nitrogen, which can also behave here as the double bond, and this carbon can now have 5 bonds. This leaving group is leaving as water. And so we have water leaving as a leaving group, so we have minus H2O here. So now we have the amino group installed. This is followed up by a nucleophilic attack by the cyanide ion here on the carbon that it's bonded to the nitrogen, this being a polar bond pulling electrons this way, so that gives us this partially positive charge right here. Should probably remove this so it's not mistaken for a mechanism arrow. Okay, fine. So that gives us the alpha aminonitrile, and then in terms of the hydrolysis of the nitrile to a carboxylic acid, that's the same reaction that we see in chapter 20 for reactions of carboxylic acids and their derivatives, and so that mechanism is discussed there. And we ultimately, though, we have an amine group attached at the alpha carbon to a carboxylic acid, and that gives us the amino acid. And again, whatever was the R group here, the alkyl group here, this is going to be the R group on the amino acid and the side chain. We can now, with some synthesis methods, we can select for the synthesis of L amino acids. So as we discussed previously, all naturally occurring amino acids are L amino acids, and their stereochemistry is such that if you draw the Fisher projection, the amine group would be positioned on the left side. And so the syntheses so far have all produced racemic mixtures because they come from a flat intermediate. So if we want only the L enantiomer, which would be true if we want to produce actual biological proteins, we'll have to find a way to resolve the racemic mixture by separating the enantiomers. We can do this with chiral chromatography. Or we can use an asymmetric synthesis that makes only 1 isomers. So clearly it's more efficient to produce the desired enantiomer, otherwise we waste half of what we've produced. And so in the synthesis of amino acids, this is an extremely important thing to do. And so there are special catalysts that can hydrogenate a bond in a certain stereochemical configuration, and that is one way of producing an L amino acid specifically. So here we have a carboxylic acid with an amine type group here. If we hydrogenate this molecule here, this example molecule with the chiral catalyst, what this means is we can direct the hydrogenation such that the hydrogen that's added ends up on the wedge as this is drawn, and then this one may be on either side here, but this nitrogen here is on the dash, or sorry, is on the wedge, and the hydrogen is on the dash. An example of a catalyst that can be used in this way is the diphenylalanine, this one here, this BINAP as it's called, and we had some examples of certain chiral catalysts which also carried this kind of a name here that could hydrogenate in a particular stereochemical configuration. And so in this case, this is going to show the production of phenylalanine. And so we have here a molecule with the double bond that is adjacent here to the amino group that has an acetate group attached as well. So this is an amide group, and the carboxylic acid. So we hydrogenate with the chiral catalyst, which produces for us a particular enantiomer, in this case with the nitrogen on the dash configuration, making the hydrogen here on the wedge. And we get 99% of this particular enantiomer. And then if we react this with sodium hydroxide and water, and then the presence of and a workup with acid afterwards, what we'll do is hydrolyze this amide, and then ultimately leave ourselves only the amine group, which is protonated due to the presence of the acid. And now the side group here is this benzyl group, which gives us the amino acid phenylalanine. So those are some ways of synthesizing amino acids. I will tell you those particular reactions, they're interesting to review because they are applications of the reactions we've already learned to the synthesis of biomolecules. However, they're not going to be my particular focus, at least in the direction of proteins. What I'd rather talk about instead is, or focus on more in this chapter, is the structure of amino acids and the structures of the peptides that are produced from those amino acids. So for example here, below we have a structure of a tripeptide here, which is known as glutathione. And in glutathione, it's composed of a glycine residue here, and a cysteine residue, and a glutamic acid residue, and a glycine residue here. And so notice that between each of these, and residue, by the way, is just a word that is used to express the amino acid in a peptide. And in each of these peptide bonds, they're formed from a condensation reaction which removes water and produces an amide from a carboxylic acid and an amine. So the side chain here for the glycine here is simply hydrogen. The side chain here for the cysteine is a CH2SH, thiol. And then finally for the glutamic acid, the side chain here is it's actually bonded on the side chain itself in this case. Instead of being bonded on the carboxylic acid group, it's bonded in the carboxylic acid group that is a part of the side chain, which can occur as well in small peptides. Not in longer ones. This wouldn't be able to continue in the same way, the long one, on ones that are longer. But when it comes to the sequence of these amino acids, the order of the sequence really does matter. It's going to give us two completely different molecules. And so, for example, if we have a condensation reaction between alanine and glycine, effectively what happens is we remove water, and then there is created a bond between this carbon and this nitrogen, and so here's that bond right here, and that ultimately gives us an amide group. This is also referred to as the peptide bond. This is the peptide bond. And so notice that in alanine, in this one we call alagly, the alanine has the amino group that's exposed on one end, and the glycine has the carboxylic acid group exposed on the other end. What we're going to see is this is called the N prime, and this is called the C prime, or the N terminus rather than the C terminus. And so the result is that if the sequence is different, if we have glycine here on the left and alanine, what we'll see is that in that case we still get an amide bond, but here it's the glycine that has the amine group exposed, and the alanine here, you can tell the alanine because it has the methyl side chain, it has the carboxylic acid group exposed. So see the difference? Alanine with the methyl has the amino group exposed on the end in this case, but in this case the alanine has the carboxylic acid group exposed in the end. And so you get two different peptides depending on the order that you put these two amino acids in. So the peptides have what's known, as I said, an N terminus on one end where the amine is exposed, and a C terminus on the other end where the carboxylic acid is exposed. It is customary to write the series of amino acids left to right such that the N terminus is written on the left and the C terminus is written on the right. And so the order of these amino acids, they do matter because the alanine here being on the N terminus is different than it being on the C terminus, right? And so we're saying something with this order left to right, that alanine's the N terminus and leucine's the C terminus, and these amino acids go in this sequence from the N terminus to the C terminus. Now remember that these peptide bonds are amide bonds, and so the CN bond has a partial double bond character due to the fact that there's resonance. So remember here we can draw a resonance arrow of this pi bond between two atoms of differing electronegativity, which gives this oxygen 3 lone pairs and 1 bond, a negative formal charge, and this carbon now has 3 bonds and no lone pairs, the carbon in the middle, and so it has a positive formal charge, and now we have a lone pair adjacent to this positive formal charge, and so we can draw this resonance arrow, and now we have a nitrogen with 4 bonds and no lone pairs, so it has a positive formal charge, and the oxygen here still has 3 lone pairs and 1 bond for a negative formal charge. And so this carbon nitrogen bond that's in the amide can take 2 different forms, because the carbon here can be attached onto the nitrogen in the S-trans configuration, as it's known here, where they're on these 2 chains here on opposite sides, or it can be in an S-cis configuration where these 2 chains are on the same side, and there's a barrier of rotation here of about 80 kilojoules per mole. This rotation is somewhat restricted because there's some partial double bond character here. It's not a complete single bond here. Peptide bonds of the peptide backbones have a preference for the S-trans configuration, and that makes sense, right? You basically have things that are more spread out, further away from each other, these groups, and so you're going to see that in the structure of the polypeptides, the S-trans configuration is the most common, and it's kind of implied by the way these are drawn. It's going to be the carbon-nitrogen bond that can experience rotation. Other backbone bonds can freely rotate, and so this backbone can kind of twist around and bend into different shapes, and that contributes to the way that a string of amino acids will fold around on each other and produce the overall structure of the protein. Now, another part of the structure of proteins is the formation of disulfide bridges between multiple cysteine residues, and this will have a big impact on the structure of the protein. Now, back when if you were with me in Chem 212, when we talked about thiols, we said they could be oxidized, and when they are oxidized, they're not like an alcohol that will oxidize to, from an alcohol to a aldehyde to a carboxylic acid. They don't oxidize in that way. When they oxidize, they lose their hydrogens here. These two hydrogens get lost, and losing hydrogen, that's oxidation too, also gaining oxygen, right? Lose hydrogen, oxidation. And when they do, we end up with a bond here between one sulfur and the other. And this is called a disulfide bridge. We talked about how this is relevant to hair curling. So if you want to get your hair chemically straightened or chemically curled, what they will do is they will expose your hair to a reducing agent, which will break these disulfide bridges in your hair, your hair is made of proteins. And then they will put your hair into a particular configuration, and then they will oxidize the proteins in your hair again to reform the disulfide bridges, and therefore hold your hair in the position that it was held into when that oxidation happened. But this has a huge impact on the structure of proteins. When the places where there's disulfide bridges between two amino acids that are far along on the chain, the chain can wrap in on itself and produce a type of structure, three-dimensional structure to it. And we'll see examples of that as we move forward in this chapter. So here's an example with a cartoon. So if you have two cysteine residues that are far along from each other on the chain, but they react with one another and they make a disulfide bridge, that will cause that will hold this polypeptide in a looped formation like this. It's an intrastrand disulfide bridge. You can also have interstrand disulfide bridges. So if you have two different polypeptides, they can link to one another by a disulfide bridge between two cysteine residues as well. So here's a few small peptides that are relevant to biology. Leucine, enkephalin, and methionine enkephalin function in the brain, and they control the pain response. Bradykinin is a hormone that dilutes blood vessels and acts as an anti-inflammatory agent. And so you can see the series of amino acids that produce those small peptides that are relevant to biological chemistry. Another important small peptide is vasopressin, which causes a reabsorption of water by the kidneys. It controls blood pressure, and it contains a disulfide bridge between two cysteine residues here. And oxytocin, another one, this induces labor and stimulates milk production, and this also contains a disulfide bridge in it. And so you may be shocked to find, okay, we actually know what these series of amino acids are, and we in fact can figure out the sequence in these small, not just in these small peptides, but even in larger sequences of amino acids that produce a large protein sometimes. And the way this is done is by breaking the amino acids at certain specific positions. And when these sequence of amino acids are broken in certain positions, we will get fragments, and then we can analyze those fragments and piece them back together to determine the total sequence of the amino acids. So one way of doing this is a so-called Edman degradation, and in the Edman degradation, we remove an amino acid from the N-terminus of the peptide as a so-called phenylthiodantoin derivative, or PTH, which is probably much easier to say. So the PTH derivative is analyzed to identify what the amino acid is. And so let's say you have a peptide, a sequence of amino acids, and at the end you have your N-terminus residue here. I'm really having a hard time. I'm trying to use a laser pointer. Okay, there we go. The N-terminus end. And so that's going to react with this PTH derivative, which contains nitrogen, double bond carbon, double bond sulfur, and the nitrogen bonded to a phenyl group, along with a trifluoroacetic acid. And that will basically hydrolyze this last peptide bond and remove one amino acid. And that amino acid will be bonded to the PTH. And here's the PTH portion right here. And so then this PTH derivative can be analyzed, and the identity of the N-terminus amino acid can be determined. And you can do this one amino acid by amino acid, one by one, until you have the entire sequence. And this works for up to 50 amino acids. After that, essentially you end up with a lot of side reactions and junk that make it difficult to determine what the subsequent amino acids actually are. So the way the Edman degradation works in terms of mechanism is that the lone pair on the nitrogen on the N-terminus N performs a nucleophilic attack on the central carbon here, the carbon here being a nucleophile, and we draw a resonance arrow here. This is a pi bond between two atoms of differing electronegativity. The electrons here go on the sulfur. Then the sulfur atoms are great nucleophiles because they're larger than oxygen atoms, so they don't hold on to that lone pair as well. And so you end up with a nucleophilic attack on the carbonyl in the amino acid, and again we have a resonance arrow of a pi bond between two atoms of differing electronegativity, which gives us, for the sulfur, it had 3 lone pairs and 1 bond, giving it a negative formal charge. But now it has two bonds, and it does have 2 lone pairs. But the oxygen now has 3 lone pairs and 1 bond, giving it the -1 formal charge. At that point, we have a proton transfer here from acid, which protonates the hydroxyl group here, and simultaneously we have the loss of the nitrogen here as a leaving group. It does get protonated by the trifluoroacetic acid, so we have a proton. This is kind of putting several steps together, but we have a protonation of the nitrogen, making it a positive formal charge nitrogen with 4 bonds and no lone pairs, which makes it a better leaving group. And at that point, the carbonyl bond can be reformed, and the nitrogen leaves as a protonated leaving group and thus leaves neutral in charge. And then finally, we have the PTH derivative, and that can go on and be analyzed, and the amino acid that is attached to the PTH portion of the molecule can be determined, and we can determine a sequential number of amino acids on the polypeptide. If you have greater than 50 amino acids, this sequence of 1 by 1, taking off each amino acid as a PTH derivative and analyzing it won't work as well. And so this is where we use the strategy that I had mentioned earlier, where we cleave the polypeptide in certain particular locations, and then based on the fragments of peptides that we get, we can put them together again and determine what the entire sequence was. Okay, so larger peptides greater than 50 amino acids, that's what this is for. And if you are given a certain amino acid called a peptidase, peptidase is referring to an enzyme that can cleave bonds in these polypeptides in certain particular positions. For example, trypsin cleaves the peptide bond on the C-terminus of an arginine and a lysine. So every time there's an arginine or a lysine in the sequence, trypsin trypsin will cleave that. There we go. Trypsin will cleave that, and you will get a series of fragments here, right? And so one ending here with lysine, one ending with arginine. And then these individual fragments can be analyzed. Another type of peptidase is chymotrypsin, and chymotrypsin will catalyze the hydrolysis of the carboxylin of aromatic amino acids. So phenylalanine, tyrosine, tryptophan, these ones will be cleaved, and you'll get a series of fragments again. And then the smaller fragments are sequenced by Edman degradation, and we determine the sequence and then piece it back together to get the entire protein sequence here. So that's going to be the end of this lecture. So in this lecture, we discuss the synthesis of amino acids. In addition, we discuss how the sequence of amino acids can be analyzed in order to determine the sequence of amino acids in a polypeptide and the nature of a protein. In the next part, we'll begin to talk about the way that peptides can be synthesized, and we will end with the overall three-dimensional structure of proteins broken up into 4 main parts, which we call primary, secondary, primary being the sequence of amino acids, which we've already talked about. We'll also talk about the secondary, tertiary, and quaternary structures. So I look forward to seeing you in that last video. Have a good one.